Good afternoon, everybody. Before I explain why we're challenging the wisdom, we're challenging how soluble dietary fibers lower blood lipids, I would like to present some facts on the current situation on diet-related health diseases, their causes and risk factors, and the strategies we are adopting in QAAFI to address these issues. And then I'll explain how using the experiments we've done in the center to challenge how soluble dietary fibers from wheat and oats lower body fat. So in developed countries such as Australia and the US, the major health-related challenges include cardiovascular disease, type 2 diabetes, and colorectal cancer. 1 in 4 Australian adults suffer from on these three chronic diseases, which are cardiovascular disease, type 2 diabetes, and chronic liver disease. This represents around 3.7 million Australians, including 1.2 million, suffering from at least 2 of these conditions. Cardiovascular disease is one of the lead causes of death and is a high health burden for all societies and mostly developing and developed countries. We have 17 million people per year worldwide die of cardiovascular disease, and mainly coronary heart disease and stroke. And in Australia, this represents 39% of all deaths. So what are the risk factors? We've divided the Australian Institute of Health has divided these risk factors into 2 categories, if you wish. The behavioral risk factors, which includes tobacco smoking, insufficient physical activity, excess alcohol consumption, and inadequate consumption of fruit and vegetables. And the biomedical risk factors, which we can measure, are obesity and overweight and obesity, high blood pressure, dyslipidemia, which include all your fat, triglycerides, and cholesterol, and impaired glucose regulation. So in the center, we've been looking at how fruits and vegetables, and I would like to include here cereals. We've worked a lot with cereals, can potentially reduce blood lipids and cholesterol, and then prevent overweight and obesity. So what are we doing in CNAFs? We are looking at the mechanisms behind how the soluble dietary fibers from cereals and fruits lower body fat. And the second approach is looking at how satiety and satiation from fruits and vegetables and nuts given as snack can make you eat less. And what we want to do in this project is to quantify fullness. So this is an ongoing project, and what also we would like to measure in this project is the metabolic rate and gastric release. I won't say too much about this project for now. But if you are interested in participating in this sensory trial, please drop me an email, and I'll be sending information about recruiting participants for this project. But if you're interested, please email me. The first project is funded by the Center for by the Center of Excellence in Plant Cell Walls. You will ask me why plant cell walls should fund such a project about lipid reduction. Well, all these soluble dietary fibers are embedded in the walls of your plant. And Mike Gidley last week very clearly told us about how the cell wall is built. So if we should know how the wall is built for us in the nutrition area to know how the body will digest the wall, and release all the bioactive component with health benefits, with potential health benefits in our gut, in our body. So this is why the center is the Center in Plant Cell Wall is funding this project. And as some of you here know, that the Uni of Melbourne and Adelaide are looking at how looking at the genetic side of the cell walls and the biochemistry of the cell walls. And the second project is funded by the Naturally Nutritious project, which encompasses QAAFI, DAF, and Horticultural Australia. And one part of what I would like to do, I've got really is close to my heart, is to translate this knowledge of fundamental science that we are generating in the center, to translate that to the general public, to the dietitians, nutritionists, food industries, and I should maybe have added the growers as well, why not? And how can we do that? We can educate as early as our children, work on policies. Working on policies, the government and agencies, bodies like SARO are doing that, are working on policies, but they take a long time. Something that I've been doing in the lab is designing and developing food products with health benefits. I've got UniQuest involved in this part of my work, and we're developing some cholesterol-reducing baked products, and we've partnered with a food industry, food manufacturer, Priestley Gourmet Delight, who will eventually market those products. I had a chance to present this work, our science, to investors and food industries at the Food Day of Future Food Day, which was organized by the Brisbane Research Council earlier this year. So I'm going to present some results, as I was saying, on this mechanism of cholesterol reduction, lipid reduction by dietary fibers. So as an introduction, and I said it before, these soluble dietary fibers, which are non-starch polysaccharides, are found in the walls of your cereal, your fruits, and your vegetables at different concentrations. If we look at oats, you will find mostly this soluble dietary fiber called beta-glucan. If you look at wheat, for example, you'll have another soluble dietary fiber called arabinoxylan. And as you can see here, there are different concentrations depending on which tissue of your grain you're looking at. So this is just a wheat grain, and just to make things a little bit things to talk to you, that in different tissues you've got your cells and you've got your walls. So those fibers sit inside, embedded in the walls. So those fibers are big polymers. You've got beta-glucan which is a homopolymer of glucose bound by 1314 linkages, and whereas arabinoxylan is a heteropolymer with xylan backbone with arabinose substituent on top of on the backbone. Those dietary fibers are viscous. They have high water holding capacity, and they can form gels. What is important is the molecular weight of those big polymers, and these molecular weight depends on the cultivar and the grain, how you process your grain. The more you process your grain, the more likely you are to damage your big polymers. Therefore, your health effect might be reduced, and storage as well can degrade, can destroy your polymers. Epidemiological studies have shown that consumption of those soluble dietary fibers from cereals and fruits cause decrease in cardiovascular disease. And from clinical and animal trials, studies have shown that oats and barley beta-glucan, wheat and rye arabinoxylan, and psyllium seed husk and pectin cause decrease in blood cholesterol and triglycerides. As I was saying before, they are biomarkers for cardiovascular disease. And this has prompted health claims on reduction of cholesterol by oat and barley beta-glucan, as well as psyllium and pectin. So the health claim reads as such for beta-glucan from oats. If you eat 3 grams or more of oats beta-glucan per day, you can reduce your blood cholesterol. However, what we don't know is how do all these soluble dietary fibers reduce blood lipid. So the mechanisms that have been proposed in literature are firstly that the fibers, as I said, they are viscous. The fibers, they bind to bile acid in the small intestine, causing an excess excretion in the feces, thereby reducing blood cholesterol. Another way they believe that the soluble dietary fibers can lower the glycemic index, thereby reducing blood cholesterol. And thirdly, the fibers, they go further down the digestive tract. They get fermented, forming short-chain fatty acids such as propionate, and those propionate will eventually cause decrease in blood cholesterol. But why is it important to understand mechanism? It is, as I said before, you can identify your cereal grain sample, better identify those grain samples to better look at your processing treatments and promote healthy eating through better designed functional foods and limit requirement for really costly clinical trials. So the proposed mechanism is the first one, that these viscous soluble dietary fibers bind to the bile acid, cause an excess excretion in the feces, and thereby reduce blood cholesterol. How does it happen? What happens? So in the body, these bile salts, the bile salts are really, or bile acids are really important in digestion of fat. They are formed in the liver, and in the liver from only cholesterol via a series of 14 or so enzymatic reaction, with this enzyme being the rate-limiting enzyme in the synthesis of the bile acid. Once the bile acids are produced, they are stored in the gallbladder, and they are released in the upper gut when fatty foods from the stomach are released in the duodenum, the upper gut. Once they've digested the food, they've done what they have to do, they are reabsorbed by the body. However, in the presence of those viscous dietary fibers, big polymers, we believe that the reabsorption of those bile acids are blocked or prevented. The consequence is that there will be a greater loss in the bile acid in the feces. This is the proposed mechanisms. How does the body react? The body, if there is an excess loss in bile acid, the proposed mechanism says that there would be an increase in the activity of this rate-limiting enzyme, which is cholesterol 7 alpha hydroxylase, which causes a decrease, because you're producing more bile acid from your cholesterol. This causes a decrease in the concentration of cholesterol in your liver. What does the body do then? Start synthesizing a lot of cholesterol, but it's smart. The body is smart. What it does, if you've got high cholesterol in your blood circulation, if it's got receptors, those LDL cholesterol receptors, which absorb, if you want, the cholesterol from the blood, thus reducing the level of cholesterol in the body. So what this is all saying is that there are interactions between the bile acid and the soluble dietary fibers in the gut. Are they really bound together? So this mechanism, this is what's been proposed. However, when you look at literature, you see a lot of inconsistencies in the report that show an excess excretion of bile acids in the feces. For example, if you look at ileostomate studies, those studies are only short term, so you can't feed someone and look at what's happening in the body over a very short period of time. It doesn't work. And secondly, there are only speculations about this bile acid binding to the fiber, and those reports haven't even measured the amount of the concentration of the bile acid in the gut when they see this reduction in blood cholesterol. Another thing is when they measure the activity of the cholesterol 7 alpha hydroxylase in the liver, when they saw an increase, but they didn't even go and again measure this amount of bile acid in the feces. They're just assuming, all right, your fibers will bind to the bile, so bile acid bind to the fibers because in an excess excretion, there are only extrapolation of what is happening. Therefore, in the past years, this is what we've been doing. We've used pigs. We've not only studied those interactions, the effect of the fibers on the lipids using in vivo models, but also we've worked on in vitro, trying to really underpin if there were a close binding of the bile acid to the fibers. And we've also done some ex vivo experiment in which we took living tissues and we looked at the diffusion of the bile acid with and without the fibers for a period of time, and using this Using chamber system, which allowed the tissue to be kept alive for around 2 hours. So in the in vivo model, we use pigs as our model because it is close, they are close to us in the lipid and bile acid in the lipid physiology. So with the model, what is interesting is that we could sample at different sites of the gut. So here you have a small representative of the small intestine. It goes like that from your duodenum to your jejunum and your terminal ileum, knowing that digestion of your lipids mostly happen towards the upper gut in the duodenum and the jejunum, and that all those bile acids I was talking about, once they've helped in the digestion of the lipids, they absorb further down the tract towards the terminal ileum. This is where most of the bile acids are reabsorbed in the body. Therefore, the model is really interesting because we can't do that with humans. We can't just take samples from different sites of the gut. So, from our in vivo experiment, one we did was using this soluble wheat arabinoxylan fraction. So we've used arabinoxylan and we looked at what was happening with the lipid digestion. So we've used 10 animals and we fed them for longer, for 4 weeks, on a diet with and without arabinoxylan. So what we saw, the blood results showed a decrease in the level of triglycerides with no effects on cholesterol. And we saw a significant difference. We saw a significant decrease in the bile acid concentration. So there was a decrease. We said, all right. So in the blood, we saw a decrease in the bile acid concentration. So what is happening in the gallbladder? So we took sample from the gallbladder, and we saw again a significant decrease. Therefore, the fiber was then sort of causing an increase, an excess production of bile acid in the body. And then we looked at the intestinal samples, and as I was saying before, we took samples from different sites. So what we saw was really, really interesting. So minus AXRF is without the fiber in the diet. With arabinoxaline in the diet, we saw a significant decrease in the amount of bile acid along the tract. So we didn't see any increase in the bile acid in the presence of the fiber. So that's going against the proposed mechanism. So that's just to show you where we've sampled. So we saw a decrease in triglyceride levels in the blood. So we went and measured, we measured triglycerides in the digestion of triglyceride, which is represented by the apparent digestibility per site. So if you look at without the fiber in the diet, with the fiber in the diet, so we have the different categories of fatty acids. But if you look at just the total fat, we see that with the arabinoxaline in the diet, there is a decrease in the digestion and absorption of your triglycerides. So less is being digested as you move along the tract. What does it mean? Less is digested, therefore less is absorbed. And this is what we saw in the blood. We saw a decrease in blood triglycerides. To summarize this experiment, with arabinoxaline, we saw a decrease in the blood triglyceride after an adaptation to the diet. This could be due to a decrease in circulating bile acids, a delay because you have less bile acid, it makes sense that less of the triglycerides would be digested, so less digested and absorbed. And we saw no excess excretion of bile acid whatsoever along the tract. And something really interesting, we talk about, and people know about the prebiotic effect of those fibers. And here we saw a change in the bile acid profile, because you've got different bile acids in your body. And in the presence of the fiber, we saw this change in the bile acid profile in such that the therapeutic bile acid, which is UDCA, which is a bile acid used as a medicine to dissolve gallstone, and we saw an increase in that bile acid and a decrease in the toxic one, which is LCA, which can cause cancer. So the probiotic effect of the fiber again here is really interesting and exciting. The second experiment we did, in this one we fed our pigs again, but this time oat beta-glucan, and we take this study to another level by looking at the diffusion, as I was saying before, of this bile acid in the living tissues. And and then this experiment I carried out with my collaborators in the University of Ghent in Belgium. So what we did is we had 10 pigs. We put 5 on a controlled diet and 5 on a beta-glucan of a high molecular weight. And we took samples at the beginning, the middle of the experiment, and after 27 to 29 days, we sacrificed the animals, and we took again samples from the different part of the gut, the feces this time, and blood as well. And then we took some tissues. We needed those tissues to put in this using chamber, and this is the display, the setup for the using chamber. And then we did also some microscopy on the tissue we took. So the results, the bile acid first, and this is representing over time. So from day 0, we started to the end of the experiment, and then in blood taken from 2 different sites, from your jugular vein and your hepatic portal vein. So what we saw was going towards a significant difference, the decrease, borderline decrease in the bile acid concentration. A borderline decrease could mean because of variation between animals, as you would know. When we look at what was happening now, what was happening in the digester along the tract, again, your different sites that's the control, that's concentration of the bile acid for the control group, and the BG group, the beta-glucan group, we again saw a very clear decrease in the level of bile acid. We never saw an increase. So, and the distal colon here represent the feed completely towards the end of your digestive tract, and we didn't see any increase. So there was over time decrease of the bile acid levels. When we did the diffusion experiment, so this is one chamber of the using chamber, and then we got a different setup like this to do replicates. So how do we do that? We place the tissue, the living tissue, in this device here, and we had a buffer solution, a mucosal buffer solution on one side, and a serosal buffer on the other side. The mucosal buffer represents your lumen, the intestinal lumen, and the serosal buffer represents your blood. So you had diffusion from your lumen towards the blood. So we put the bile acid on one side of the chamber, and we measured the diffusion of the bile acid with and without the beta-glucan in the mucosal side of the chamber, and across tissues taken from both groups, from the control group and from the beta-glucan group as well. So what we saw was there was slower diffusion. This is your GDC, one bile salt we use in this experiment, was slower across tissues with the beta-glucan from the beta-glucan group. What is this saying? That if I take tissues from the tissue from animals on the beta-glucan diet, that tissue, the bile acid was diffusing slower. And then what we saw, again, so we added, as I said before, we added beta-glucan in the chamber as well. And then again, we saw a significant decrease compared to the control group. We saw a significant decrease in the diffusion of the bile acid. So this is saying something has happened to your intestinal tissues. Looking at the cholesterol levels, so in the blood, over time, we saw at the end of the experiment a significant decrease in total cholesterol compared to, this is your control group and this is your beta-glucan group, and a decrease in LDL cholesterol, so your bad cholesterol. So decrease in the blood, and when we look at what's happening along the digestive tract, so this is your control, and this is your beta-glucan your beta-glucan concentration from the beta-glucan group. So what is this saying is we have a decrease in cholesterol in the blood, but in the digester you have an increase. It is saying that the fiber is preventing the cholesterol from being absorbed, right? So it's sitting in the gut and to be expelled, to be excreted. And what was intriguing when I saw those results was this drop. I was thinking, what's happening? Why is this drop here? It's still going. Would have thought it might have gone and the cholesterol be excreted, but why did it just drop? Then we quantify. I looked at it closer, and I saw the formation of this corpus tunnel, which is a non-absorbable sterols. What is it saying is that here, the bacteria transform, so the fiber promotes good bacteria and transform your cholesterol into corpus tunnel, preventing further cholesterol from being absorbed. And that was really exciting. That's really great. When we look at the bile acid profile now at the different sites with the different diets, we see, as I was saying with the first experiment, we saw the same thing in the second experiment. We saw a decrease in the toxic LCA and an increase in the therapeutic UDCA. So to summarize, with the oat beta-glucan, we saw a decrease in blood total and bad cholesterol after an adaptation period, the diet. This could be due to a decrease in circulating bile acids, prevention, the beta-glucan prevented cholesterol and bile acid active uptake. The transformation of cholesterol into corpus tunnel and non-absorbable neutral sterol. And again, we saw these prebiotic effects of the oats. It changes the profile of the bile acid profile into the increase promote the good bile acid, UDCA, and decrease the toxic one. What we think is happening, so when you've eaten a fatty food, those fatty food, your cholesterol and your triglyceride enter your upper gut. The gallbladder excrete bile acid. The bile acid help the bile acids help the digestion and absorption of your triglycerides and your cholesterol, and then they are themselves reabsorbed toward the end of the gut, as I said, not the end of the gut, but towards the terminal ileum or the small intestine. And when we look at what's happening in the liver, and this is an area that's starting to pick, it's not just simple, it's all happening in the gut, but everything is related. If you look at what's happening in the liver, the bile acids, they are ligands. They are ligands for this receptor, the nuclear receptor, fascinoid X receptor. The bile acid bind to this receptor and causing a suppression of the conversion of cholesterol into bile acid. So that's the normal state of the body. The body, as I said, is smart. It doesn't want to produce a lot of bile acid because they can be toxic, right? They are toxic. So at all times, there is a suppression of this conversion of cholesterol to bile acids. In the presence of the fiber, as I said, they are viscous, they are gluey, they form sort of a network around your lipids, around your cholesterol. This gets into the upper part of the gut, and somehow we believe that the body, the cells, sense less of the fat, doesn't send this. If you've got a fatty meal, they just say, oh, there is not too much fat in the body, in the gut, in the lumen. So what happens is from the gallbladder, only small amount of bile acids are released. And this, talking about the sort of network around the lipid globules. This is what we've done. This is my PhD student who's finished now has and published this paper looking in vitro what is happening to the lipid droplets in the presence of the fiber. So on blue is the fiber, and those so that's beta-glucan, and those are lipid droplets. In this black and white image, we can see the sort of a network and completely trapping the lipid droplets, prevented digestion from happening. So this is what we think is happening here. And if what's up, so the body sends less fat, therefore increases this suppression of the formation of bile acid from cholesterol. Therefore, we don't have an increase in the formation of bile acid. And this is what we saw in the results. When we look at the images from the microscope from TEM, we saw, so this is your small intestinal epithelium, we saw an increase in the activity of the goblet cells. Goblet cells are cells which produce mucin, and so this is, if there's an excess excretion of mucin, a production of mucin in the presence of the fiber, this could explain a decrease in absorption of nutrients in the presence of fiber. And we saw as well a decrease in the height of the microvilli, again restricting absorption. So the fibers, the soluble dietary fibers, have potential health benefits in preventing cardiovascular disease by decreasing blood cholesterol and or triglycerides, The mechanism should, however, be revised. And then, as I said, we need to look at more, look closer at these nuclear receptors, because those receptors link the bile acid and the lipids and the lipid metabolism. The effect of the soluble dietary fibers are seen after an adaptation period on the diet, on diets. And then we saw the soluble dietary fibers as being a prebiotic, which is interesting, increasing the good ones, the good bile acid, and decreasing the levels of the bad ones. And here I would like to thank Margaret and Hannah for helping in the media release of the second work we've done. The take-home messages would be the oats and arabinosyl and reduced blood cholesterol and triglyceride after an adaptation period with implication towards promoting good gut bacteria. However, the mechanism should be revised and thus help to be revised to better monitor eating habits and diets, and thus help prevent hyperlipidemia, obesity, and cardiovascular disease. And lastly, indulge. Keep eating your oats and your wheat. I would like to thank Mike Gidley for his support. It's been full on, and he's been really supportive, and there have been a few challenging moments, and he's always been supportive. And I'd like to thank all those who worked on the pig experiment, the first experiment I presented, and a PhD student, and all my collaborators in Belgium, and some overseas students who work on the project as well. And thank you all for your attention.